Hello, I'm Mirela Delibegovic and I'm a professor at the University of Aberdeen, and I'm delighted to tell you today about our CSO-funded project on the development of a sensitive and high-throughput antibody assay for SARS-CoV-2. This is a collaboration between the University of Aberdeen, Vertebrate Antibodies Limited, AI Biologics, as well as NHS Grampian and NHS Tayside. I would also like to take this opportunity to thank the Share Bio repository for providing us with the crucial samples that were required to develop this assay in the first place. So I'll start off by describing the COVID-19 serology test development at the moment. And the current tests use either the N or the S antigen of the virus. So what this means, whether we're using ELISA or a lateral flow, is that we would get a positive or a negative response when we're looking at antibodies. However, what these assays don't take into account is that we have a heterogeneous humoral response in population. And what I mean by this is that 5% of population are either spike positive or nucleocapsid positive, and therefore we wouldn't be capturing accurate data from other patients. We also know that accumulating mutations reduce sensitivity, and also that subdominant epitopes can mask the signal. And the big problem with the assays is that specificity is compromised due to high homology with other seasonal coronaviruses. And this comes into play when we look at this slide here in our table 1, where we use the Share Bio repository samples. So here we had 100 samples from pre-COVID, which is pre-2018. And our COVID-19 samples from ICU units from NHS Grampian and NHS Tayside, as well as the US population. So here we expressed, this is the region of the virus, either SARS-CoV2 N protein or the S protein. We can see if these are individually expressed that we are getting some antibody readouts, 85% or 72% in the case of S-based ELISAs. However, we are also getting these false positives in the pre-COVID samples, so there is a cross-reactivity. If we combine the N and S proteins together, we do get 87 or 88% positivity in regards to antibodies against SARS-CoV-2. But also, we can see here, if we use pre-COVID samples, that this comes up to 43% positivity, which is a false positive. And if we express and use the N protein of a common cold, so a seasonal coronavirus, we can see that we get 100% positivity when we're looking for pre-COVID samples or the COVID-19 samples. Therefore, there's a really urgent need to develop better and more accurate and sensitive assays. So this is where our technology comes into play. So obviously the perfect scenario would be that we would be using the AI technology to help us do the immunogenicity profiling of the SARS-CoV-2 antigens, and that we would then use all the different parts of the virus to actually develop our assay. So we want to identify immunodominant epitopes, and these are in our boxed regions here. And ideally, we would want to fuse these epitopes together on a display platform and then do the ELISA assay. The advantages of this ideal assay design would be maximum sensitivity as well as specificity. We would achieve this by eliminating the subdominant as well as cross-reactive epitopes. Another advantage is that the test combines the sensitivities of multiple SARS-CoV-2 antigens. So as we can see here, we've got S, N, M, and open reading frame. This would also overcome SARS-CoV-2 genomic variability, and also it would be highly flexible. And when I say highly flexible, as mutations come about, we can then include these in our assay development. However, when developing these, one faces two challenges. The first challenge being the identification of these immunodominant epitopes, which is not straightforward. And secondly, there was no affordable technology available to combine and display these multiple epitopes on this scaffold that we would want to use. So the way we overcame the first challenge is by the use of the artificial intelligence application Epitope Predict. So Vertebrate Antibodies Limited for over 20 years has been generating antibodies for different species. and every time a new antibody is generated, data is inputted into the AI software, which is a machine learning software. What this means in case of SARS-CoV-2 is that Epitope Predict allowed us to recognize the immunodominant epitopes and the corresponding antibody classes as well as subclasses. And this is achieved by the machine learning capability of the software. And this was giving us predictive accuracy of about 92%. Second challenge, as I said, is the expression technologies being expensive and inappropriate. So if we think of a current peptide arrays, we have different peptides attached to the plate. We would have our serum sample and fluorescent antibody binding. So the cons of this approach is that chemical synthesis is very expensive for these peptides, cost is highly limiting, and there's a limited success rate. There are also additional steps required to retain native conformation, so for example, biotin, and new settings are required. In addition to that, expression systems range in their sensitivity. So for example, multiple epitopes have to be used to achieve this high sensitivity in assay that is required, but it's very difficult to produce these in E. coli due to their small size. So here's an example of a Comasi stained gel where we used different epitopes using expression systems, and what we can see here is that one gets poor expression and poor stability. We get quite a bit of degradation as well as aggregation. So the solution to the whole problem was the epitope antigen, or our epitogen technology. And the epitogen technology was developed in this project to display single or multiple epitopes on a scaffold for direct use as an antigen. And this allowed us to get both high specificity and high sensitivity. And we achieved 100% success rate. Our costs were very low, and we achieved high yield. Also, everything is achieved through bacterial expression, so this allowed for easy scale-up. So as I said, the Epitope Predict SARS-CoV-2 model, AI software, was used to predict these immunodominant epitopes that would then be tested in the lab and then fed back into the system. So this is presented in a model here. So this is our Epitope Predict AI software, which would suggest different epitopes. These would then be expressed on our scaffold, which was also developed during the project. We would do the quality verification by routine ELISA. We would then have to eliminate those epitopes that were decreasing the sensitivity, go back to the AI software and redo everything again. So the key to the epitogen technology is really the scaffold, and the scaffold was developed in the project as well. The scaffold had to be inert and provide basically an anchor for our proteins of interest. It had to be stable. We had to be able to produce it in high yield and purify it easily, and also had to have functional domain for binding and screening. So the reason that this was really important is exemplified here. So in November or December 2020, we were starting to get patients in the ICU with different mutations, and this is a comparison using an antibody test that we were developing in kind of using the reference epitope, which is the original virus, versus the samples from the patients who had a Y144 deletion. So we can see here in black the antibody response in the patients who had the original infection with the original virus. However, if one were looking for antibodies in the patients that were presenting with a Y144 deletion, we can see that there is absolutely no antibody response produced. This is obviously not the case, likewise with other mutations such as P681H. So this patient 208 here was infected with the reference stain, and the mutation evades the antibody response. So it's pretty clear that better assays are required. So our epitogen technology was basically allowing us to use the scaffold and then develop and bind to it different epitogens. And each epitogen could have about 5 epitopes. So in theory, we could then express 30 different epitopes in the same reaction. And this would allow us to have high sensitivity and high specificity. So I don't really want to go through this quite a complicated diagram, but what I want to stress here is that this allowed us to really use all different parts of the virus to kind of achieve this high sensitivity. So we could use the spike protein, we could use the nuclear capsid protein, as well as the membrane protein in an open reading frame, and design through our AI software different epitopes that could be fused together. So we would have them either singly or fused together that we could test against each other. So for example, if we concentrate here on the nuclear capsid part of the SARS-CoV-2 virus, we could see that there were different predictions in regards to immunogenicity of different parts. We could express these individual epitopes on our epitogen platform. So we can see here expression of different epitopes. And then we could test each one of these epitopes against our serum samples. So the samples obtained from COVID-19 patients or our negative controls, which are the pre-COVID samples. And we could see here, for example, with N6, so this is our epitogen N6, that we have high antibody readout for this part. So what we would do next is then fuse these different parts together to create a composite epitogen, so N6. So we can see here when they're individually expressed on a ponzo gel, on a Comasi gel, I apologize. And this one is the fusion of all of these together. So 5 different epitopes into one single epitogen. We call it N6, and N6M is the one that has mutations also incorporated within it. So now when we are testing positive samples as a percentage positivity, we can see that only if we incorporate all 5 epitopes together into one single epitogen that we get over 90% sensitivity in the assay. Likewise, we did the same with the spike-based antigen. We expressed each one individually, tested again against our serum samples, plasma samples, tested them individually, but also all combined together. So 5 different epitopes into one epitogen called S6. And we can see here when all are combined together that we get over 90% sensitivity. Now, here we can see that we've talked about the N6 already, so the epitogen that detects the end part of the viral particle, and also the S6, which is the spike part. But we also said that the beauty of the system was being able to add in the membrane protein as well as the open reading frame. So likewise, these were created into one single epitogen, giving us the sensitivity of about 92% at this point of the assay development. And really kind of coming to the SARS-CoV-2 genetic variation, what we know is that mutations as they come about are going to affect the sensitivity of the assay. So what we were able to do by developing this technology is to include these mutations here. So if we concentrate on the spike part, if we're looking here, which is about 97% sensitivity, if we add the mutations in and then fuse these together, S6 plus S6 mutations, we can see that we get really high sensitivity. Likewise, we could include all the reported mutations in the end part of the particle so that we can basically combine the N, M, and open reading frame and the mutants and the spike mutants to come up with our epitogen universal assay, which achieves over 98% sensitivity. Another thing that we were able to do and develop through our project is looking at the sensitivity over time. So in this table here, we can see days post-PCR. So these are our patients that had blood collected at days before 8 to 14 days, 15 to 21 days, up to 28 days, post-positive PCR, and over 28 days. And what we can see here is as the patients have been affected with the virus and as the time passes, we reach a peak at 22 to 28 days where we are really getting the clinical sensitivity of around 100%. After 28 days, this starts to decline slowly. And this is the combination of all the parts of the virus as well as the mutations. And the beauty of the whole assay is that as the mutations come about, we can just include them in the assay. So for example, when Omicron came about, we just had to go back and add in those mutations into the assay to maintain the sensitivity. So this was, as of January 2022, this was the state of play with the different assays that we developed for this CSO-funded project. So we developed the universal SARS-CoV-2 antibody assay, which passed phase 2 validation and independent evaluation by the National Institute for Biological Standards and Control on the 17th of November, as well as the differential SARS-CoV-2 antibody assay. And this is a really nice one because it also allows, in the one single test, to differentiate between vaccine-induced and infection-induced antibody response. This is really important going forward to understand the prevalence in the population and how well we are protected against the future pandemics. We're also trying to develop, and we are currently developing, mucosal SARS-CoV-2 assay and the immunity SARS-CoV-2 assay. And we're also taking part in the establishment of the second World Health Organization international standard for immunoglobulin to SARS-CoV-2 and reference panel for variants of concern. So thank you very much for your